Okay, now is the white screen visible to everyone? I hope it is visible to everyone. Okay, so
Yes.
now let us revise what we have been doing so far. Okay, I would just let you know what step we have learned about the life cycle of this plasmodium. Okay, once it get entry into your liver sinusoids, it further get matured into schizont or miront. Then after nuclear division, some 800 to 1000 cryptomerozoites are released, and these cryptomerozoites then again attack new liver cells. Okay, and these cryptomerozoite then further enters into new liver cells and again start out the same stuff. Okay, so the very first round of division, right from its entry into liver cell till the formation of this cryptomerozoite, well, some people term it as pre-erythrocytic cycle or first phase, and then the subsequent or remaining repetition of release of these metacryptozoites into the liver spaces, and again reinfection. It is termed as this exoerythrocytic schizogony. Okay, so now at the end of this exoerythrocytic schizogony, what happens is that we have two type of metacryptomerozoites. Okay, we have mega metacryptomerozoite and we have micro metacryptomerozoites. Well, this is what in short I would like to tell you that this is what has happened. Okay, in previous lecture we have dealt with the detailed step wise, development wise, what happens in that parasite morphology and everything. We have learned about all these stages of the parasite in this stage of life cycle. Okay, we also concluded our discussion with something called as relapse. Okay, since this stage
Sir,
of this
can I,
parasite. Hello.
sir, can I, sir, can I interject once? I'm having some trouble with viewing your screen. Uh,
Is
so I
it the
sent
same with
you.
everyone?
We can see,
No.
sir, normally.
Yes, other can see. There might be problem with on your side. Maybe some connection
Yes, I
problem,
understand.
or
Yes.
and anyhow, I am recording this lecture, so you would
Right, right.
eventually get the lecture, right?
Okay, okay, sir. Yeah, I actually shared the screenshot with you on WhatsApp. You can actually check if you want once. So it's like the entire thing is kind of, twisted. Okay,
The
I'll try connecting to my phone. I think that will be better. All right.
okay, okay, ठीक है, please,
Okay.
please try.
Sorry, sir. Sorry, sir.
Okay,
Sorry, please continue.
okay. So we stopped our discussion with relapse of malaria, right? So the relapse is said to be occurring due to the parasites that are present in the liver. Okay, and since they are present in liver, they remain hidden from the immune system, as well as they are inaccessible to the antibiotics, or you can say the anti-malarial drugs that you provide. So whatever malarial drugs that are usually given, they do not have any tendency to kill the parasites that are hidden into the liver. Whatever drugs that we are giving, they only attack the parasite which are present in RBCs. Okay, so now in case of Plasmodium vivax, these relapse can occur at say for example at least for 2 years. That means when the person is cured from malaria and he is infected with that Plasmodium vivax, even after 2 years, there is a strong possibility that whatever parasites that were present in the liver of that individual, they can again come up and cause malaria in that individual. Okay, we have also discussed that in case of Plasmodium malaria, this relapse period is far more greater and it can extend up to several years. While in case of Plasmodium falciparum, the relapse do not occur because the liver phase is self-terminating. Well, this particularly means that if an individual is infected with Plasmodium falciparum. Once the parasite gets entry into liver, all of the parasitic load would get entry into your RBCs, and none of the parasite would be left behind into liver to cause the relapse. Well, that is what the self-terminating liver phase means. Okay, so now what happens next is that this after these micrometa cryptomyrozoite and megameta cryptomyrozoites are formed, they now have tendency to leave liver and invade the red blood corpuscles, or what we call RBC. Okay, so this is called as erythrocytic schizogony. So erythrocytic schizogony is the phase of life cycle of Plasmodium in which they infect something called as the erythrocyte. Okay, so now in erythrocytic schizogony, these micrometacryptomyrozoite that are liberated by the erythrocytic phase enter into bloodstream. They establish contact with RBCs and enter them to start erythrocytic schizogony. It has been reported that there is a variation regarding the selection of erythrocyte in different species. Okay, say for example, Plasmodium vivax attacks only young and immature erythrocyte. On the other hand, Plasmodium malariae invades only old erythrocyte, while Plasmodium falciparum has tendency to attack immature, mature as well as old erythrocyte. Okay, so the entry of the parasite into the erythrocyte marks the very onset of malaria. Okay, so in the next phase, what happens now in RBC? So whatever we are going to study now, the next phase of the life cycle of this parasite, we are going to see whatever happens in our erythrocyte now. Okay, now we have left liver and our parasite has entered into RBC. Okay, so now once this micrometacryptomyrozoite feed on the cytoplasm, it gradually grows and reaches about half the size of the erythrocyte. Okay, so this green color that you see over here is your micrometacryptomyrozoite. So this micrometacryptomyrozoite, once they get entry into RBC, okay, after they get entry into RBC, what they do is that they increase in size and they usually become half of the size of RBC. Okay, well, this step is usually carried out by consuming all of the materials that is present inside your RBC. Okay, so initially, as you can see over here, when this micrometacryptomyrozoite gets entry into your RBC, the vacuole is somehow smaller. Okay, now the vacuole as well as the size of this parasite increase inside this RBC, and due to the increase in the size of this vacuole, the nucleus of your parasite is then pushed toward one of the side. Okay, so now due to this pushing of the nucleus to one side, a large vacuole develops. Okay, and this gives the appearance of signet ring, and hence this stage is called as signet ring स्टेज. ओके, अब ये किसके जैसा दिख रहा है? एक अंगठी के जैसा दिख रहा है, इसलिए ये स्टेज को क्या कहते हैं? पैरासाइट को वी कॉल दिस स्टेज एज सिग्नेट रिंग स्टेज. ओके, now the further parasite becomes amoeboid. Okay, when the further growth of this parasite takes place, it no longer remains spherical, it becomes a kind of amoeboid in nature. Okay, and now it occupies the entire space in your RBC. Okay, so the enlargement of parasite, you can say it increases up to 2 to 3 times of the initial micrometacryptomyrozoite in which it has infected the RBC. Okay, so now what happens is that at this stage, okay, amoeboid stage, it then grows and occupies entire RBC. Okay, so what happens now at these stages, this parasite expresses the enzyme called as heme polymerase. Okay, so this heme polymerase, okay, well, what this heme polymerase, sorry, it expresses the enzyme called as heme polymerase. Okay, so what happen is that this heme polymerase is responsible for converting heme into a substance called as hemazoin. Okay, so prior to expression of this heme polymerase, some of the proteolytic enzymes are also expressed in this parasite. Due to activity of this proteolytic enzyme, the hemoglobin is broken down into the protein component of hemoglobin, that is globin, and whatever the remaining portion of hemoglobin, that is the heme, it is it is present in form of a pigment called as hematine. Okay, so what happens is that heme is supposed to be highly toxic for the survival of this parasite. So using the enzyme heme polymerase, what this parasite does is that it convert this hematine to something called as non-toxic form of hematine called as hemozoin. ओके, तो बात समझ में आ रही क्या? सबसे पहले क्या होगा? When this Shizon stage is achieved after the amoeboid stage of our parasite, and it occupies the entire RBC, it started to release some proteolytic enzyme. Okay, so ये proteolytic enzyme क्या करते हैं? It break downs the hemoglobin into two parts, the protein part, the protein component, what we call it as globin, and the remaining component, which is heme. This heme is somehow toxic to this parasite, therefore it is converted into hemazoin. Okay, so this hemazoin is non-toxic and thus parasite can survive. Okay, so furthermore, whatever globin portion has been created due to proteolysis of this hemoglobin, that is digested, and the remaining portion, that is heme, is left in form of hematine. तो ये heme polymerase क्या करेगा? It would convert that hematine into hemazoin. Okay, so along with this, some small pigmentation, also called as the Schaffner's granule, or some yellow dots are formed on the surface of the erythrocyte, जैसे कि यहाँ पर दिख रहा है आपको. These are called as Schaffner's granules, or some yellow or orange granules are also produced. Okay, so once
Sir,
this, yes.
Can you please tell again about the Schaffner's granules? What are those exactly?
Okay, when this we will come to that in a while. Okay, so these Schaffner's granule are nothing but some orange or yellow small granular bodies which are produced as a side product of the metabolism that is happening in and around the parasite. Okay, बात समझ में आ रही है आपको? So these
Yes, sir.
Schaffner's granules, आगे अपन इसके बारे में बात करने वाले हैं. Okay, so now once it reaches this schizont stage, the nucleus of the schizont now divides and redivides to form at least 12 to 24 nuclei. Okay, now this 12 to 24 nuclei come to lie at the periphery of this schizont. Okay, so what happens further is that each and every of this nucleus would now surround itself with a piece of cytoplasm to form small unique nucleated body called as merozoites or schizozoites. Okay, so the merozoites show a definite arrangement within an erythrocyte around the residual cytoplasm, which contain melanin and metabolic waste. So now you can say that this Schaffner's granule, melanin, and other metabolic waste, or you can call it them as toxins, are produced by these parasites. Okay, so at this stage, we say the parasite has attained the Roset stage. Roset stage किसे कहेंगे अपन? The divided nuclei comes at the periphery of the schizont, and they started to gather around some of the cytoplasm around itself. Okay, so by this stage, the erythrocyte gets weakened. Okay, and they burst around, releasing all the toxic material that has accumulated within it. Okay, so all the merozoites in different erythrocytes break out at the same time. So this results in a diagnostic chill followed by a very high fever. Okay, so in spleen, kidney, and liver, the residual cytoplasm and melanin are eaten up by phagocytosis. Okay, so this merozoite once again enter into new erythrocyte, and the erythrocytic schizogony is repeated all over after 48 hours. Okay, so this cycle, in case of our what was our model organism for plasmodium? We were talking about plasmodium. काय के बारे में था?
Why?
प्लाज्मोडियम. ये काय के बारे में पढ़ रहे हैं अपन? Do you remember that?
Plasmodium vivax.
Plasmodium vivax. Okay, तो Plasmodium vivax में क्या होता है? Well, the entry of this micrometa cryptomerozoite till bursting out of this merozoites. Hello, who is talking? I can't hear.
अच्छा नेहा बात कर रही.
हाँ नेहा बोलो बेटा. Shall I proceed?
डिस्टर्ब हो रहा है शायद से.
Okay. Okay, I will proceed. Anyhow, we are recording this thing, so I don't think anyone would miss out anything. Okay? ठीक है. So,
Okay, sir.
हाँ, the time taken by this micrometa cryptomerozoite to get entry into RBC and further development and release of merozoite or schizozoite, it usually take 48 hours. And therefore, every 48 hours, when this cycle is repeated, well, it results in diagnosis. Everyone is being, सबको सुनाई दे रहा है बेटा, आपको सुनाई नहीं दे रहा होगा तो भी ये रिकॉर्ड हो रहा है, and you would get the copy of recording. So if there is disturbances in your signal or network, so please be patient. I will give you the recording. Okay? ठीक है, तो ये जो साइकिल है, राइट फ्रॉम द एंट्री ऑफ योर पैरासाइट इनटू आरबीसी and bursting out, it requires 48 hours. So this cycle would repeat after every 48 hours. That would result in the typical chills as well as high fever, and therefore we say that these are the typical symptom of malaria. Okay, the fever would come after every 48 hours, and due to release of all this toxic material from the erythrocyte, when everything burst out, it would create that typical chills. So chills with fever with periodic nature, well, the doctor would then suggest ki the person should go and do that malarial test. Okay, so these are typical these are typical symptoms of malarial fever. Okay, so this is how things happen in your erythrocytes. Okay, now this repetition of erythrocytic schizogony, that is entry into the erythrocyte, followed by different stages of development, multiplication inside the RBC, rupture of RBC, and again entry of the newly formed schizozoids or neurozoid into new RBC. Well, this repetition of erythrocytic schizogony would result in tremendous increase in number of plasmodial parasite into the blood. Okay, so since these parasites ruptures the RBC when they come out of the RBCs, okay, so this would eventually cause anemia in the infected person. So this condition usually result in death of the host and ultimately that of the parasite. Okay, so the parasite the parasite has also to face the problem of shortage of food and that of the resistance of the host. Okay, because this parasite has been present for a longer time in liver as well as now it is present in RBCs also. Therefore, there are very strong chances that these foreign bodies might have been recognized by our immune system, right? And therefore, the change of the host becomes essential for the continuation of this life cycle. Okay, so this is marked by the replacement of schizogony by the sexual phase in which gametocytes are produced from these schizozoids or merozoites. Okay, so now adaptation or you can say conversion of merozoites to gametes or gametocyte is referred to as one of the parasitic adaptation of this organism. Okay.
Sir, I have one question.
Yes.
That is when you were talking about chills, that
Yes.
is the symptoms.
Hmm.
So we have often seen that there is a remission or there is a relapse in malaria. Malaria, like there's a gap of a few weeks or few months between a few days between successive chills. So वो क्यों होता है? So मतलब
यही तो
what
बोला
does?
मैंने कि वो साइकिल
Okay.
रिपीट होने को 48 आवर्स लगते हैं,
अच्छा अच्छा. Okay. अच्छा.
राइट?
All right.
So
Okay. All
whatever
right, sir.
parasites that are released from the previous cycle,
Mhm.
they would get entry into new RBC and again
Okay.
develop themselves into merozoites or schizozoid. तो ये सारा cycle होने को it would take around 48 hours. तो 48 hours तक
Okay.
फिर कोई symptoms नहीं रहेगा. Why? Because all the parasites are present in that RBCs.
हाँ. Right.
Okay,
Right.
and when after
Okay.
48 hours all the developmental stages are completed and there are lot of or you can say large number of parasites present inside the RBC, then they would burst out at the same time.
Right.
And
Right.
when they burst
Okay.
out, they release all the toxic material that has been accumulated inside. That would result
Mm-hmm.
in subsequent chilling and high fever.
Right. सर और एक क्वेश्चन था, ये जो जब वो बर्स्ट आउट होता है, जब वो लाइसिस
Hmm.
होती है,
Hmm.
तो उसके बाद वो सिम्टम दिखाने में कितना टाइम लगता है? मतलब वो लेटेंसी का पीरियड क्या है? मतलब आफ्टर बर्स्टिंग एंड एक्सप्रेशन ऑफ सिम्टम्स, इसके बीच में कितना टाइम
नो
पीरियड
नो
जाता
नो नो,
होगा?
वो बर्स्ट होते ही वो टॉक्सिक मटेरियल बाहर आ जाता है और शुरू हो जाता है आपका
ओके
सिम्टम्स.
ओके, इमीडिएटली होता है. राइट.
चिल्स एंड
ओके.
फीवर्स हर 48 आवर्स बाद आएंगे इफ दिस स्पीशीज ऑफ प्लाज्मोडियम इज इन्फेक्टिंग दैट इंडिविजुअल.
राइट. ओके सर. ओके. थैंक यू.
Okay. So now due to all these changes, okay, so once और कहाँ पर थे अपन, we were talking about the parasitic adaptation, right? It has to face few difficulties now. सबसे पहली difficulty क्या रहेंगी उसे कि ये काफ़ी दिनों से वो host के body के अंदर रहेगा. Well, there are chances that the immune response might have been developed and there might be immune cells like macrophages and everything that might be on hunt of this parasite. दूसरी चीज़ क्या है कि it would require a lot of RBCs, healthy RBCs, to infect for the new merozoites or schizozoids that are formed. Okay, but due to previous cycle of bursting of this RBC, the number of RBC would eventually go on decreasing. राइट. सो इसके आगे अभी दो चैलेंजेस है. एक पहला वाला इम्यूनिटी से बचे रहना, और दूसरा shortage of food. Why? Because the erythrocyte that were already present in that host body are now somehow on the depleting stage, right? So in order to cope up with this, what happens is that some of the merozoite, after entering the erythrocyte, they do not develop into schizont. बात समझ में आ रही है आपको? अगर ये वाले जो मीरोजोइट और शिजोजोइट है, व्हेन दे गेट बैक इनटू इरेथ्रोसाइट्स, they do not develop into this schizon stage, but they develop into gametes or something called as gametocytes. Okay, so this is called as one of the parasitic adaptation. Okay, so what happens is that after entering the erythrocyte, they do not develop into schizont, but undergo a gradual development to form large and compact round bodies. These are now known as gametes. इसे क्या कहते हैं अपन? Gametes, or we also call them as gametocyte. Okay, in later stages of development, these stages can be morphologically distinguished from one another. Okay, so at this particular stage in your bloodstream, there would be some RBCs that would be having the parasite in schizon stage or all of the other development stage, while there would be some RBCs that would be having these gametes. Okay, now these gametes can be distinguished into two types. Some gametes are larger, we call them as megagametocytes, and some of the gametes are smaller, we call them as microgametocytes. Okay, so this
Sir,
huh?
I had a doubt.
Yes.
The merozoites or the schizozoites, they undergo anemia, starvation, and form gametes, or they infect RBCs and they don't become a schizont stage, but they again form gametes, right?
Gametes, yes.
Like I was confused. So yeah,
ओके,
okay.
तो ये गैमोन्स और गमीटोसाइट क्यों तैयार करते हैं? क्योंकि ऐसे ही चलता रहा तो होगा क्या?
Mhm.
That would result in complete anemic nature of the disease. There would be no RBCs and eventually the parasite would die,
Yes.
right?
Mm.
So in order to continue its survival, it has to find out some another strategy. So instead of
Okay.
infecting new RBCs and further developing into schizon stage, what do it do
Mhm.
is that it develops into some another morphological form that we call it as gametes.
Okay. Okay.
ओके, तो ये गैमोन्स
Yes.
का आगे डेवलपमेंट होकर दो टाइप के गैमोन्स आपको दिखाई देंगे. व्हाट वी कॉल इट एज माइक्रोगेमेटोसाइट्स एंड मेगागेमेटोसाइट्स. माइक्रोगेमेटोसाइट्स आर स्मॉल एंड मेगागेमेटोसाइट्स आर लार्ज. ओके,
Yes.
बात
Yeah.
समझ में आ रही?
Yes,
ओके,
sir.
सो एट दिस स्टेज इन ब्लड स्ट्रीम, आप ब्लड स्ट्रीम में जो कुछ भी आरबीसीज रहेंगी उसमें कोई आरबीसीज में माइक्रोगेमेटोसाइट रहेंगे, मेगागेमेटोसाइट रहेंगे, या फिर कोई आरबीसीज में शिजोन के अलग अलग स्टेजेस के डेवलपमेंट भी रह सकते हैं. ओके, okay, now let us talk about these microgametocytes and megagametocytes. Okay, as I have already told you, these macrogametocytes मतलब बड़े वाले, macro या mega. Okay, these megagametocytes eventually form large you can say gametophyte. Okay, so these macrogametocytes or megagametocytes are female gametocytes. फीमेल गमीटोसाइट्स यानी क्या? ये जो सेल्स है इससे फीमेल गमीट आगे तैयार होने वाला है. ओके, एंड दीज आर वेरी न्यूमरस इन द पॉपुलेशन. Okay, so they do have small and compact nucleus, and this nucleus is situated toward one side of the cell. Okay, on the other hand, this microgametocyte, okay, which eventually further form male gamete, well, these are smaller in size, these are less in number. Okay, and the nucleus is larger than that of मेगागेमेटोसाइट, और इनका न्यूक्लियस जो होता है दैट इज सिचुएटेड एट द सेंटर ऑफ cytoplasm. So based on the morphology of these two parasite the cellular structure, you can identify कि इसमें से कौन सा मैक्रोगेमेटोसाइट है और कौन सा माइक्रोगेमेटोसाइट है. Okay, macrogametocyte or what we call it as megagametocyte would further develop into female gamete. Microgametocyte would further develop into male gamete. Okay, बात समझ में आ रही है सबको? Is it clear now?
Yes, sir. Yes.
Okay. So now these gametocyte would remain in the erythrocyte for several weeks. Okay, तो ये जो गमीटोसाइट है, माइक्रोगमीटोसाइट या मेगागमीटोसाइट, ये आपके इरेथ्रोसाइट में कुछ हफ्तों तक भी रह सकते हैं, बट दे कैन नॉट डेवलप फर्दर अनलेस दे आर ट्रांसफर्ड फ्रॉम ह्यूमन होस्ट टू अनदर होस्ट, दैट इज फीमेल एनोफिलिस मॉस्किटो. Okay, if the mature gametocytes are not transferred to specific host, they would degenerate and die. Okay, so at this stage it is necessary that some another healthy mosquito comes and take a blood meal, so that these RBCs with these gametes or microgametocytes or megagametocytes, you can say, then they would get transferred into the female Anopheles mosquito, and further development would occur in the female Anopheles mosquito, that is the vector host. Okay, so all you can say from this discussion is that the gametocytic phase, okay, is initiated in human host, but it cannot complete this phase into human host. It needs to be transferred into the vector, that is female Anopheles mosquito. Okay, is it clear now? Asexual phase
Yes, sir.
completely happens in human.
Yes, sir.
Okay,
Mhm.
सेक्सुअल फेज का जो गमिट फॉर्मेशन होता है वो इनिशिएट तो होता है आपके ह्यूमंस में, बट एंड होता है उसका आपके मॉस्किटो. Okay, okay. So now,
Yes.
what would be the next stage? The next stage would be the female Anopheles mosquito would again go and take a blood meal, and along with the blood meal, all of these different stages of parasite would get sucked into that mosquito. Okay. So this is how things happen in case of human. Okay. So abhi let us study what happens in the female Anopheles mosquito or vector. Okay, so this is what we have all discussed so far. Okay, well now let us see the life cycle in female Anopheles mosquito. Life cycle of Plasmodium in female Anopheles mosquito. Okay, so what happens at this stage is now the thousands of the parasite in various stages of development along with a gametocyte enter the alimentary canal of female Anopheles mosquito. Okay, which sucks the blood from human beings suffering from malaria. So in the stomach of Anopheles, all the remaining stages of the parasite are digested except the gametocyte, as they can withstand the action of the digestive juices that are released in the digestive tract of the mosquito as such. ओके, तो यहाँ पर आरबीसी जब उसने ब्लड मील लिया तो well there were RBCs which were having lot of other developmental stages of the parasite, but during the digestion what happens is that everything is digested except these gametocytes. Okay, why? Because these gametocytes has ability to withstand whatever digestive juices that are present in the digestive tract of that female Anopheles mosquito. Okay, now let us study the gametogenesis, that is how this microgametocyte and a megagametocyte would result in formation of male and female gamete. ये प्रोसेस को क्या कहते हैं अपन? We call it as gametogenesis. Okay. So once these microgametocytes and megagametocytes get entry into the stomach of the vector, the further development of gametocyte takes place in the stomach of intermediate host, which is usually a cold-blooded invertebrate. Okay. So the lower temperature is favorable for the development of gametocytes of Plasmodium. Okay. So the nucleus of well, let us first study how the male gametocyte develops first. Okay. So this male gametocyte would develop from microgametocyte. Okay. The nucleus of your microgametocyte undergo rapid division and produces about 4 to 8 daughter nuclei, जैसे यहाँ पर दिख रहा है आपको. ठीक है. So these daughter nuclei then migrate on the periphery of the cell. Okay. And subsequently 4 to 8 long slender outgrowth from the main body of microgametocyte are given off from the point where the nuclei are situated. Okay, तो अभी क्या होगा, यहाँ पर होने के बाद में after this stage, after the division of this nucleus and the migration of this nucleus towards the periphery of the cell, there is process called as exflagellation. तो ये exflagellation में क्या होगा, कुछ 4 से 8 आउटग्रोथ तैयार होंगे आपके माइक्रोगेमेटोसाइट में से. Okay. And this outgrowth में eventually whatever the nucleus were there, they would migrate into that outgrowth and the cell would then separate themselves. Okay. And finally these microgametes are formed. At this stage you can see the process of exflagellation. Okay, दिख रहा है यहाँ पर. These are small thread like slender outgrowth. Okay. और इसमें फिर आगे न्यूक्लियस जाएंगे and then they would separate and they would form the microgametes, and these microgametes are also referred to as male gametes or sperms. Okay, बात समझ में आ रही?
Yes.
Yes, sir.
Okay. So now as compared to this, this development of female gamete is quite simpler. इसमें ज़्यादा complication नहीं होता है. Okay, the development of this micro gamete or mega gamete from macrogametocyte is comparatively simple. Okay, the nucleus of a macrogametocyte, that would result that would eventually divide and result into something called as the polar bodies. क्या होगा, आपका जो मेगागेमेटोसाइट है उसका न्यूक्लियस है, उसका डिवीजन होगा, डिवीजन होने के बाद में क्या होगा, कुछ पोलर बॉडीज तैयार होंगे. Okay, so when these polar bodies are extruded, we say that the female gametocyte has been matured and you can say the gametocyte has been formed or the ovum is formed. Okay, I would repeat it once more. The nucleus of a megagametocyte would divide to initially form a few polar bodies, and these polar bodies are then removed out. Okay, after the removal of polar body, we say the ovum or female gamete is formed. Okay, तो जब ये मैच्योर ओवम तैयार होता है, the nucleus of this ovum moves towards the periphery, and that point of cytoplasmic projection is developed, which is called as the fertilization or reception cone. तो जब ये ओवम तैयार होता है, फिर क्या होता है, इसका जो न्यूक्लियस है वो पेरीफेरी पर आता है एंड सम रिसेप्टिव स्पॉट इज फॉर्म एंड दिस रिसेप्टिव स्पॉट इज कॉल्ड एज द रिसेप्शन कोन. Okay, and it is said that in this area fertilization will happen. Okay, तो बात समझ में आ रही है आपको? Do you get how this male gamete is formed from microgametocyte and ovum is formed from megagametocyte? Okay, it is quite simple. So the next logical step would be what? You might have learned it. एक बार फीमेल गमिट तैयार हो गया, मेल गमिट तैयार हो गया, that would result
Think of
into
fusion.
fusion.
Yeah.
That is called it as fertilization. Okay, so now we have got this male gamete and we have got this female gamete. Well, let us talk about fertilization for a time being. Okay, fertilization as well as the embryonic development. Okay, so these there are several micro gametes, or what we call it as male gametes. Okay, since they are very slender, they can move by their lashing movements towards the macrogamete or ovum, but of these several male gametes, only one can penetrate through the fertilization cone. Okay, now nuclei of the both gametes will fuse to form a diploid nucleus, something called as synkaryon. Okay, तो fertilization होने के बाद में क्या होगा? It would form a diploid nucleus. It is called as synkaryon. The cytoplasm of both the gamete also fuses completely and a single cell zygote is formed. Okay, so this fusion of two dissimilar gamete is called as anisogamy. Okay, dissimilar gamete यानी क्या? Male gamete is different, or you can say is morphologically different from the female gametes. You can differentiate between the two gametes, therefore we call it as dissimilar gamete. So in zoology,
Yes.
when two dissimilar gamete are fusing together, such a type of phenomenon is called as anisogamy. ठीक है? And since
Yes.
there is nuclear as well as cytoplasmic fusion, the process is called as syngamy. ओके, तो ये फर्टिलाइजेशन का दूसरा फीचर क्या है? इसमें न्यूक्लियस के साथ में दोनों गमीट्स के साइटोप्लाज्म भी फ्यूज हो रहे हैं, एंड देयरफॉर सच अ टाइप ऑफ फर्टिलाइजेशन इज कॉल्ड एज syngamy. Okay, so now this spherical zygote, which is first inactive, soon becomes active and grows into an elongated vermiform motile eukainate, or it is also called as vermicule. तो पहले क्या होता है? ये zygote initially जब तैयार होता है, well that is usually सुस्त होता है वो, but after some time it gets activated, it changes its morphology, okay, and it becomes something called as eukainate, or we call it as vermicule. Okay, so this eukainate starts gliding
Sir,
and wriggling.
eukainate or
Vermicule.
Vermicule.
Vermicule. Okay, so this eukainate start gliding and wriggling movement, and with the help of its tapering end, it pierces into the wall of stomach. अभी क्या है? इसका morphology change हो गया. It has become fusiform, or what we call it as vermiform, and using its tapering end, it can get its entry into the stomach wall. Okay, so these eukainate which are the eukainate which are not successful in penetrating the gut wall are eliminated out along with feces and host. तो ये सब कहाँ पर हो रहा है? अपना मॉस्किटो के स्टमक में हो रहा है. तो जरूरी नहीं कि जो कुछ भी जायगोट तैयार होंगे वो सारे के सारे सक्सेसफुली आपका जो मॉस्किटो का स्टमक वॉल है उसके अंदर पेनिट्रेट होंगे. इट इज नॉट लाइक दैट. जो कुछ भी बेचारे अनफॉर्चुनेट जायगोट्स है जिनके eukainate पेनिट्रेट नहीं कर पाए, well those are then washed along with the feces of the host. होस्ट यहाँ पर कौन है? आपका मॉस्किटो. ओके, सो आगे ये eukainate का डेवलपमेंट कैसा होता है ये अपने को पढ़ने का है. Okay, so the successful zygotes or ookinete lie below the outer epithelial layer of the gut wall and assume again the spherical stage. Okay, so होगा क्या कि अभी ये ookinete कहाँ पर चले जाएगा? It would go and attach itself to the outer epithelial layer of the gut wall. Okay, और gut wall में पहुंचते ही it would again become spherical. Okay, so once it again becomes spherical, a thin membranous elastic cyst wall is secreted partly by ookinete and partly by stomach tissue of host around the ookinete. Okay, तो ये क्या करेगा? वो वॉल में जाने के बाद में it would develop a very elastic cyst wall. Okay, so this encysted ookinete is now called as oocyst. Okay, pierce the stomach wall. Okay, दिख रहा है ये? तो ये oocyst का क्या होगा? Now this oocyst grows about 5 times its original size and at the expense of stomach tissues and coelomic fluid. तो स्टमक वॉल में जाने के बाद में जब ये oocyst तैयार होगा और ये oocyst वहां पर क्या होगा? इनलार्ज हो जाएगा. ओके, इनलार्ज कितना बड़ा हो जाएगा? ओरिजिनल साइज से इट वुड ग्रो इन साइज ऑफ विल से फॉर एग्जांपल 4 to 5 times एंड द प्रोट्रूड एंड इट स्टार्ट्स टू प्रोट्रूड आउट ऑफ द स्टमक वॉल. वो ऐसा बाहर दिखने लगेगा यहाँ पर, जैसे यहाँ पर दिख रहा है आपको. दीज आर द ओरिजिनल पिक्चर्स. ओके, एंड दिस इज जस्ट डायग्रामेटिक रिप्रेजेंटेशन. ओके, at this stage we call them as oocyst. इसे क्या कहते हैं अपन? oocyst. आपका जो ookinete है, once it get entry into the stomach wall, it got increased in size and now we call it as oocyst. इसे क्या कहते हैं अपन? oocyst कहते हैं, or simply cyst. So now the cyst the cyst intrudes from the surface of the stomach wall and it's been easily seen in these histological slides. Okay, so it is said that a single infected stomach of female Anopheles may contain some 50 to 500 protuberances on the outer surface. Okay, तो वो जो मॉस्किटो है उसके स्टमक में कितने रह सकते हैं? फ्रॉम 50 to 500 ऐसे oocyst नॉर्मली रहते हैं. ओके, तो आगे क्या होगा? oocyst में आपके पैरासाइट का फर्दर डेवलपमेंट होगा. ओके, सो वॉट एवर फर्दर डेवलपमेंट दैट हैपन इनसाइड दिस oocyst इज कॉल्ड एज स्पोरोगोनी. क्या कहते हैं उसे? वी कॉल इट एज स्पोरोगोनी. स्पोरोगोनी यानी क्या? The development of the parasite inside the oocyst is called as sporogony. Okay, so now let us talk about what happened in sporogony. Okay, so the oocyst attain full size and enter the phase of asexual multiplication. तो अभी यहाँ पर क्या शुरू हो जाएगा? asexual multiplication शुरू हो जाएगा. The nucleus of each oocyst repeatedly divides. Okay, अभी क्या होगा इसका? जो पहला डिवीजन होता है, people say कि भाई the first division in the oocyst is meiosis and later जो कुछ भी होंगे वो सारे क्या होंगे? माइटोसिस होंगे. So इससे आपको अभी एक बात समझ में आ रही होगी कि आपका जो प्लाज्मोडियल पैरासाइट है, it remains in diploid stage only from the formation of zygote till oocyst. बाकी के सब स्टेजेस में it would remain in haploid nature. Okay, there should be no confusion in this anymore.
तो सर मतलब बेसिकली
Sir, can you repeat?
ह्यूमन होस्ट में तो फिर डिप्लॉयड स्टेट होता ही नहीं है.
डिप्लॉयड सेल होता ही नहीं, हैप्लॉइड ही होता है.
Right. Okay.
सर इससे पहले आप जो बोल रहे थे वो लास्ट पार्ट क्या आप रिपीट करेंगे?
कि देखो आपका जो पैरासाइट है वो डिप्लॉयड स्टेज में जायगोट तैयार होने से लेकर oocyst तक ही डिप्लॉयड स्टेज में रहता है.
ओके.
All the other stages are in haploid stage.
Okay. Hmm.
Okay.
Yes.
Okay. ठीक है. So once this happen, oocyst में स्पोरुलेशन नाम का एक प्रोसेस होगा जिससे क्या होगा, वॉट एवर द oocyst उसके अंदर जो कुछ भी फर्दर डेवलपमेंट होगा, सो ईच न्यूक्लियस would now get surrounded by piece of cytoplasm to form sporoblast. तो पहला डिवीजन कैसा होगा, मियोसिस होगा, उसके बाद में सारे के सारे माइटोसिस होंगे. तो ये जो माइटोसिस होने के बाद में जो कुछ भी न्यूक्लिया यहाँ पर तैयार होंगे, they would gather around the piece of cytoplasm around itself and the so called body is called as sporoblast. उसके क्या कहते हैं अपन, sporoblast. Okay, so this sporoblast will undergo the process called as excistation, in which numerous sickle-shaped sporozoites are formed and those are released out. Okay, so finally sporozoites are liberated when the cyst wall ruptures into the hemocoel of the host. These free sporozoites that are released, okay, they are highly motile, they are highly infective, okay, and due to their motility they reach over the salivary gland of the mosquito. They penetrate the wall of salivary gland and enter them, right? The entire development from gametocyte to sporozoite takes in 2 to 4 weeks. तो गमीटोसाइट से लेकर स्पोरोजॉइट रिलीज होने तक को कितना टाइम लगता है, 2 से 4 वीक्स, डिपेंडिंग ऑन द टेम्परेचर एंड अदर फैक्टर. बट नॉर्मली ये सारी चीजें 2 से 4 हफ्तों के अंदर में हो जाती है. सो नाउ when this infected female Anopheles bites a healthy person along with its saliva, these numerous sporozoite would get matured, okay, and those are released in this hemocoel. They would migrate to salivary gland, और salivary gland से कहाँ पर जाएंगे वो, when this mosquito takes next blood meal, she would transfer our parasite in sporozoite form into new host.
Yes.
Okay, so in this way the life cycle is completed. Okay, do you remember we begin with this stage, okay, the inoculation of the parasite in sporozoite form into the bloodstream. Okay, कितना टाइम हो गया बेटा?
3:55.
Oh, well, I think we should stop. अभी जो अगला पोर्शन है, ओके, टाइप्स ऑफ मलेरिया एंड कंट्रोल ऑफ मलेरिया, ये अपन नेक्स्ट लेक्चर में कंटिन्यू करेंगे. ओके, आई वुड टेक अ ब्रेक, राइट?
सर वो कितने बजे स्टार्ट करेंगे अभी?
अभी 15 मिनट्स बाद. ओके,
ओके ओके.
Okay, sir.